Hello guys, welcome to CCI iGEM's YouTube videos. My name is Andrew and this is my second video, and today I'll be explaining bacterial transformation. Um so okay, let's get started with the introduction on bacteria. So first of all, what is bacterial transformation? What is the premise for this video? So this is when the genome in the bacteria is altered, and this is very important because bacteria often they take in uh, genetic material from other bacteria through horizontal gene transfer and transformation processes. And this is important for bacterial genetic diversity and to ensure that bacteria are not all the same and that they can adapt. So this is for evolutionary purposes. It's one of the things that bacteria do. So a bit of an introduction on the anatomy of the bacteria. So there are some basic structures. So the capsule is sort of just the outer layer of the bacteria, which is a polysaccharide layer. And I'll sort of explain the differences in the capsule structure for like different types of bacteria. But essentially it's consisting of a polysaccharide that sort of serves for protection. But as you can see, if you look under a microscope on like a cell plate, you can see bacteria moving along the surface because it's also adhesive. And then there's the pili, which are just sort of like tiny filaments on the surface of the capsule. And this is sort of helps with like docking bacteria and helping to like sort of funnel this plasmid transfer process. Flagella is a tail-like structure that actually is different ways. It has I think I'll explain in depth later, but it's essentially allows bacteria to have this motility. It allows it to move, and it consists of a hook and a rotor and a tail and all of that, a filament, and yeah. And then it's very different compared to animal cell. For instance, DNA is not contained inside a structure like the nucleus. It is in a um, sort of general area as a nucleoid in the middle of the cell, but it is not sort of in a set area. There's no membrane surrounded organelles. And it has a single chromosome, which is essentially just a circular thing. Also, it contains plasmids, which are short circular strands, which also play a role in sort of bacteria genetic programming. So the flagella, here's a bit about its structure. So it's very hollow, it's a filament, and it's made of a protein called flagellament, which is a type of cytoskeletal structure. The cell slides or a video from CCI iGEM should explain more about what a cytoskeletal element is, but essentially it's made from flagellum, hence its name. Uh, and so there's a periplasmic area between its membranes in which stores a ring, sort of, and various proteins that helps it anchor to the cell. And it has a hook which allows it to move its tail. So it's actually not like moving by itself, it's because the hook is rotating. So sort of a bit about gram staining and gram testing. So essentially scientists use a technique in order to sort of identify what type of bacteria they're dealing with. So there's a gram positive and gram negative bacteria, each with its own respective differences. First is when you do a technique called gram staining. When it's negative, it generates a pink stain. And when it's gram positive, it's purple. And the purple is just the presence of peptidoglycan, which is a complex starch polymer that is found in gram-positive bacteria. And it's not present in animal cells or gram-negative bacteria. And sort of gram-positive bacteria, they secrete exotoxins, which helps to protect the bacterium and can induce disease in sort of foreign species that come close to it. Hence why you hear about so many bacterial infections and all of that. And then gram negative is essentially without, and there's definitely more gram negative bacteria. It's far more diverse, and it has a lipopolysaccharide on its membrane in place of the peptidoglycan, acting as a sort of defense mechanism. So plasmids are short, non-chromosomal DNA molecules that can replicate itself and have genes that program sort of some of the functions within the bacteria. Um, and sort of it's a vector, so it can be transferred and altered, and these changes actually are reflected in the bacteria in real time. Um, and it's sort of in a circle. So, and again, sort of bacterial genomes are circular and they replicate as so. So bacteriophages are just viruses which replicate inside of bacteria, and some archaea, and they don't really harm humans because again they're targeting bacteria, and they sort of engage in the lytic cycle, which is different from the lysogenic cycle in that the lysogenic cycle sort of acts as a parasite, sort of using it, or commensalism or whatever, but it essentially uses the lysogenic bacteria use the host as sort of a replicating center, like a factory where you continuously do it for long periods of time. But in bacteria, bacteriophages undergo the lytic cycle, which essentially is like you use the cell and all this machinery to replicate yourselves, and then you just break up the cell and destroy it in order to release yourself. And but it helps with sort of transferring genes, so that's a good thing. So here are some steps in transformation. So horizontal gene transfer. So again, the horizontal gene transfer is what we're going to be mainly explaining about today. There's three parts of it: transformation, transduction, and conjugation, each of which are examples of this transfer. And it sort of allows for a means of transferring genetic material between bacteria. And it's really crucial for evolution. This allows bacteria to evolve and to get unique genomes because, again, bacteria don't sexually reproduce like humans or other organisms, and plus, thus their sort of offspring are identical to them. So this is their way of staying unique and sort of evolving. Um, so the first step is transformation, and this is when the DNA from a dead organism is transferred to a section of the new living bacterium. And this happens when your the bacteria is sort of dead, either from a bacteriophage or from like naturally, the cell sort of bursts and releases these plasmids that can then penetrate through the membrane of other bacteria and sort of change genome a bit. Transduction, again, is through bacteriophages, which have their own genomes, and that can sometimes it donates bacterial DNA from other um, bacteria that were involved in the lytic cycle in order to become a part of its plasmid instead of like just replacing you or destroying the cell. This happens sometimes. Finally, conjugation is when there's a direct physical connection between these bacterial cells, allowing for the direct movement of genes from one cell to the other. And this usually involves pili. Again, these are filaments on the surface of the bacteria and allows for like a transport. Transposons, these are jumpy genes and jumpy genes of sorts, because uh okay, so they're sort of like encode enzymes for themselves, which allows it to move, and this helps it to move in horizontal gene transfer, and the genes sort of program for enzymes which cut themselves, and then it cuts its target sequence and sort of reseals itself, like sort of basically it's like it has its own cutters which cuts itself away from its original sequence, cuts the new sequence apart to give it space, and then pastes itself using glue in it. And this helps with things like reverse transcription, right? So you want to sort of create DNA from RNA, and this helps. And restriction enzymes sort of helps with this like reverse transcription thing where you go from RNA to DNA. And this is, in this case, restriction enzymes are DNA cutting enzymes, uh, and they're usually used for protective purposes in bacteria. So if your bacteria is penetrated by a bacteriophage, these restriction enzymes can help to destroy the bacteriophage's genome of sorts, viral genome, making it immobile or like sort of useless. And they make staggered cuts called sticky ends, and these can also help to isolate specific parts of the plasmid and to start like the transfer process where you take a specific segment and to move it to someone else. And humans actually utilize this process directly from observing these bacteria, and they found that they could use this restriction enzyme, which have very specific regions which it targets within a sequence to extract it through sort of splicing the sequence. And there's sort of many applications of this. This can be used to replicate plasmid DNA and to from manipulate bacteria. Scientists can manipulate bacteria by manipulating these transfer functions and sort of restriction enzymes in order to produce specific proteins and substances that might be helpful to humans. And this is actually the premise of iGEM and our team's projects. So check that out if you're interested. And to modify the bacteria itself. So here you see a cell line or culture with a lot of bacteria. This is actually a lot. So the liquid's actually, just for a fun fact, this is like a type of bovine solution, I think, with like media and some bovine extract. And these, it sort of like makes it into a substance where it can cultivate cells through providing with nutrients. This has a lot of cells, right? But anyhow, and it's, yeah. So anyways, some quiz sort of questions that you can choose to pause this video and answer. So what is the horizontal gene transfer method involving a dead organism? So I'm gonna give you 5 seconds to answer this, or you can pause it. Oops. So this is transformation, and again, remember this is when like a bacteria either dies through apoptosis or is sort of destroyed in a lytic cycle by a bacteriophage. So it's transformation. Sorry, I showed the answer to the previous one. Hopefully you didn't see that. But if a bacterium is gram stained and the color is purple, is it positive or negative? I'm gonna give you 5 seconds to answer this, and you can pause. It is gram positive. Remember, the purple indicates the peptidoglycan is present. I can never pronounce that word in the cell membrane, and thus it gives the purple color instead of the pink for gram negative. Okay, finally, what are the circular pieces of genetic material in bacteria called? I'm gonna give you 5 seconds again, and you can pause this video. These are plasmids. Again, remember, plasmids are unique bacterial sort of fragments of DNA, and they're circular. And yeah, that concludes our presentation today. Thank you for watching this video, and I hope you watch more if you enjoyed this. So yeah, and subscribe. So yeah.